Good afternoon, everybody. I'm recording this lecture, which corresponds with gastrointestinal tract. So let's start by sharing my PowerPoint. All right. So we're going to talk about digestive system, and we will do a lot of comparison between the different species and how the digestive or gastrointestinal tract changes or varies between different animals. So first of all, what is digestion? We need to define what digestion is. Well, it's a series of sequences by which the food is broken down, and we will see that this is important in the sense that we need to diminish the size of the food particles in order to increase the attack surface of enzymes responsible for digestion. And of course, food is going to be broken down and it's going to be chemically converted in a way that can be easily absorbed by the cells, mainly lining the intestine, in particular the duodenum in most species. And also allowing these smaller particles of food to be utilized by the body and maintain the vital functions. So we will distinguish in digestion a series of well-defined chemical different processes. So first, a mechanical action, which is mainly complied by chewing and intestinal contraction. So by chewing, we are mechanically breaking down the food into much smaller particles, and intestinal contraction, especially peristalsis, and in the case of a rumen, the contraction of the dorsal and ventral sac will contribute with that, as well as the reticulum and another of the four stomachs. So this will be really important. One of the things we normally do, especially with cattle, is we control in the flank of the animal contraction of a rumen. That gives us an idea of how healthy the gastrointestinal tract is when we assess that. So then we have a chemical action, which is mainly done by the hydrochloric acid secreted in the stomach. We will see this with a little bit more detail. And that allows, among other things, to actually decrease the pH to 2 to 3, which is kind of quite low, in order to the enzyme to work at their correct or right pH. Each enzymes throughout the intestinal tract and gastrointestinal tract has an ideal pH for fulfill the chemical actions. And also the other component that plays a very important chemical action is the bile that is secreted, synthesized by the liver and secreted by the gallbladder. And this is important for especially the digestion of lipids forming the micelles, because as we know, fat and water, they do not actually mix up together very well. So in a way to increase the absorption and digestion and the enzymes to attack mainly lipases, to attack those fat feedstuff, we need actually to form the micelles in order to also diminish the size of these particles and improving the surface of attack. And then we have an enzymatic action, which is mainly done by the enzyme. I mean, there's quite a lot of enzyme. We will be talking about some of them in this lecture. We'll have another lecture more specific about that. And the other ones in charge of degrading macromolecules to the more basic components in order to be absorbed by the intestine and the rest of the gastrointestinal tract. And they are, these enzymes can either be produced by the organ, for example, the pancreas will be a good example, or by microorganisms. And here we have cellulases. As we know, it's an enzyme that is not synthesized by the host, but synthesized by the microbes that are located in the rumen, and that, of course, contributes to the digestion of cellulose and hemicellulose of plants. So if we take as an example a general diagram of a digestive tract, we start with the mouth, which is continued by the esophagus, which actually connects the mouth with the stomach, then the stomach followed by the duodenum, site of most of the absorption of different nutrients, followed by the jejunum, the ileum, and the cecum, and the large intestine, and ending up in the anus. And if we simplify as a concept the structure of the digestive tract will be like a long tube, and if we were able, if this was possible to dissect completely the whole digestive tract, and we put it on a straight line, we would be able to see light on the other side of the digestive tract. So it is basically a long tube, and of course folded many times and adopting different shapes to for a better function, and of course to adapt to and be inside the body. So it's a tube. We can simply say that it's defined as a tube that extends from the mouth to the anus. First, we have the mouth with the lips, the teeth, the tongue, and the salivary glands, each of them, of course, fulfilling a specific and important role, mainly for apprehension. We will see that the apprehension of the different feed will depend on the species of animal considered. Then is followed by the esophagus, which mostly has a transport function. Then we have the stomach, where most of the enzymatic and chemical digestion occurs, followed by the small intestine, which normally we will describe three main regions in order from the mouth to the anus, which is going to be the duodenum, the jejunum, and the ileum. And finally, we have the large intestine, mainly composed of the cecum, colon, and finally the rectum, which is the last portion of the digestive tract. Let's start with the mouth, and here, as you can see, it's fundamental for prehension. And I included this picture of a cattle showing the tongue and a scythe at the bottom, which, because sometimes we use as a reference or as an illustration, as a graphic way to describe the prehension in ruminants, particularly cattle, I'll say much more than sheep, is that with the tongue, the tongue in cows actually plays a role like a scythe. I mean, grabbing a bunch of grass, and with the teeth and the upper part, manage to cut this grass, and that's how they actually apprehend. So they are not very selective in the way they ingest their feed. If you want, as a different with the horse and with the sheep, and if you ever, which are much more selective, meaning that they can actually specifically select certain types of grass or plants, which is not the case for cattle. Okay, and this is something you can observe on the field, especially when you see horses grazing. You can see how mobile, what a kind of a lot of movement and important is in prehension, the upper lip in the horse, upper and lower lip as well, but mostly the upper lip. So this prehension is mainly done by the lips, the tongue, and the teeth, and contributes, of course, the mouth was the main site for mechanical digestion, especially this reduction in particle size of feed is done by the teeth, and we will see how the different teeth shapes change in accordance to the species considered, and of course that has to do with the diet and how different type of teeth contribute to this digestive process. Also in the mouth, there are other roles that are fulfilled by this organ, which is wetting, keeping the feed wet. That contributes and improves digestion. It also acts as a lubricant, mainly by the saliva, the mucus present in the saliva. Saliva is about 99% water, and the other 1% is mucopolysaccharides, which actually called mucin, which contributes to this kind of slippery and lubricant function of the saliva, so for the animal being much better to swallow. And of course, in some species, there are some enzyme addition, like amylases, in the case of humans and, for example, the dog. Then in the mouth, it's also where the flavor, taste, and smell, so the sensing of these organoleptic properties of a feed are going to be perceived by the animal, mainly by the tongue. We have all these papilla distributed within among the tongue that actually allows the animal to perceive the flavor and the taste and smell, and the sinuses. And of course, it is the site of a formation of what we call the bolus, which is this kind of formation of this spherical, we can say, shape of the food particles that is much better for the animal to swallow. It's going to be much easier. So let's have a look at the dentition. Let's have a look at the teeth. They are, of course, involved in mastication or chewing of food. And they are classified based on their location and function, okay? So we have first the incisors, also sometimes referred as cutters. They actually pick up and manipulate the food. Then we have the canines, which can be defined as the piercers. They actually tear the prey. They are most of the time curved and sharp, which gives them the ideal shape to fulfill their function, especially in carnivores. Remember, carnivores, of course, they feed mainly on animal tissues, other animals' tissues. Then we have the premolar, which actually pierce the feed during the early chewing. So it's kind of a transition before reaching the molars, which are mainly adapted for chewing and crushing. And the molars, in the case of a ruminant, will be fundamental for a thorough crushing of the plants, especially the plants' walls, which, remember, are mainly made of cellulose. So the animal really needs to crush it very thoroughly, very well, so it can be attacked by the cellulose and hemicellulose enzyme, cellulase and hemicellulase enzymes secreted by the microbial population in the rumen, so it can actually be digested, right? And cell plants and cellulose and hemicellulose is quite hard, so that's why it's so important the function of the molars in crushing that thoroughly that food. And of course, then we have the process of rumination. We're going to talk a little bit about that, which is an extension, we can say, of the crushing of the molars done by the animal when he's normally resting. And also, there is in the cell walls of plants, we will find silica, which contribute to the hardness of the structure of the plant structure. And of course, it is implied in a lot of teeth wearing, which is the main cause of discarding animals in extensive grazing system, where when the animal has really worn down teeth, we need to take them out of the field. If not, sometimes they can even die during the winter. So in this diagram, we can see a comparative diagram of 3 different species: carnivore, herbivores, and omnivores. In that case, as an example, the men. And we can see how the different shape of the teeth changes, and the structures of teeth that are homologs between different species are colored with the same color, right? They are colored with the same color. So we can have also here, this diagram is actually comparing the different structure and disposition of the teeth according to the diet of the animal. We have the very long and curved canines in the dog. Then we have, and the herbivore, for example, the molars will be kind of adopting a more flat shape, a more looking like a mill for grinding the cell plants really well and making them ready to be digested. And of course, the human as an omnivore and showing different shapes and different sizes of teeth, adapting to the different components of and in relation to a very varied diet that normally humans and omnivores have. So this is a series of pictures showing the teeth in cattle. So you can see how we go from the temporary set, also called milk teeth or milk dentition, and we move towards, as the animal ages, from the first pair of permanent teeth, 2 teeth, then the 4 teeth, 6 teeth, and 8 teeth, what we call a full mouth animal, and that's how we normally determine the age of cattle. And in this table, we can see the appearance of a different teeth in cattle and the age they actually are showing. Normally, cattle are less than 2 years old when they change their deciduous teeth to the definite teeth. At 2 years, normally we will have the appearance of the 2 permanent incisors. At 4 years, we will have 4 to 6 permanent incisors. And then all the teeth when they are over 6 years of age. And about, it says they're around 12 years old, but I can tell you that that changes and varies a lot with the animal, and more than anything, it varies a lot, not with the breed, but it varies with the diet. In those animals that are, for example, grazing more soily, more sandy soils, like for example, this region in North Carolina, normally cattle will have higher or more important wear of the teeth, and sometimes the culling rate is higher. So this is something important to be checking. And of course, at around 12 years of age, we expect some of the teeth to be missing already. The other component of a gastrointestinal tract, which is important, are the salivary glands. Normally, most species have a sublingual glands, and they have also submaxillary glands and the parotid gland, normally represented by one on each side. And in the glandular stomach, we will mainly define or distinguish 3 main regions. And the size and the distribution of this region will change with species, but generally speaking, we can distinguish these 3 zones within the stomach. The first one is the cardiac gland region, which is mainly in charge of secreting mucus, followed by the proper or fundic gastric region, and finally the pyloric region, which will end up connecting with the duodenum or with the small intestine. So the cardiac is mainly a place in a region of the stomach where mainly mucus will be secreted, and the main role of this mucus will be actually to protect the stomach lining from the much more acidic gastric secretions we will see in the fundic region. And in the fundic region, we have the presence of the parietal cells, which are responsible for secreting chloride acid, which contributes to lower the pH down to 2 to 3, which is, if you think about it, is quite low. Remember, pH maximum is highest is 14. And this actual acid environment is the one that contributes to the activation of pepsinogen into pepsin. And pepsinogen is secreted by another type of cell we will find in this region called the Chief cells. And it's mainly an endopeptidase, so meaning for the breakdown of protein. And we also find renin, which is an important enzyme, mainly also proteolytic, but it's very important, especially in young lactating animals, because it contributes to the formation of curdle in the milk, which is necessary for these animals to actually digest and absorb the nutrients from milk of a mother. And then we have the pyloric sphincter, which actually controls the transit from the stomach to the small intestine, to the duodenum. And one thing I wanted to say here, that we have 2 sphincter, the cardiac sphincter, which is the one that connects the esophagus with the stomach, and the pyloric sphincter, which actually controls the passage of digester from the stomach to the small intestine. And both are really important because during stomach contraction, they actually block the passage of food either for the duodenum or to the small intestine. So when we look at the structure of a small intestine, we realize that it's an anatomy that is actually designed to, which main function will be to increase the surface area for digestion and absorption. So the whole anatomy of the small intestine, it is actually, we can say, has been created for that. So the folds will actually increase the surface area of the intestine 3 times. The villi will do it with 10-fold times the surface area. And finally, the microvilli, more than 200 times the surface area. That's what they actually help increasing. And of course, the microvilli here, we're talking at the cellular level. So as you can see, the villi and the microvilli, actually, they are all fulfill the fundamental role of increasing the surface area for both digestion and absorption. And this is crucial for digestion for the animal. Here we have another diagram which actually goes from the macro anatomy to the microanatomy, showing how the different first curves of the intestine contribute to increase the length. And then we consider that normally an animal will have several feet length. I mean, of course, depends on the size of the animal, but several feet long intestine, followed by the villi and the microvilli, which are actually lining the epithelial cells in the intestine. So in the small intestine, as we said before, we distinguish three main regions: the duodenum, the jejunum, and the ileum. That is the main role of the small intestine is crucial because it's where most of the digestion and absorption occurs of nutrients. And also in this part or region of the gastrointestinal tract, we have the accessory glands. And by accessory glands, we mainly refer to the pancreas, which is the organ of the gland in charge of secreting mainly buffers, which are fundamental for the pH control of the pH in the different regions of the gastrointestinal tract. Mainly this buffer component is bicarbonate. And then, of course, pancreas secrete enzymes. And also, don't forget, it is a fundamental gland for the secretion of hormones as important as insulin and glucagon, actually regulating the glucose levels in blood, in the organism. Then we have the liver, which is responsible for synthesizing and secreting the bile, which was fundamental, as we said, for forming a colloid, with the lipid forming the micelles, contributing for the digestion and absorption of lipids. So we have the large intestine, we have a cecum, which of course its size will vary a lot with species, and we will see that. The colon, the same situation, size will change a lot in relation to the species, animal species considered. And finally, the rectum that actually is responsible for excreting all the solid feces to the outside. Cecum and colon of the omnivores normally present bands of smooth muscles called tenias, which actually tenias means cestworm because they look pretty much like flatworm, like cestodes. And they cause, by this special structure, they actually help for the formation of pouches, giving to the cecum and colon a sacculated appearance like sacs, which you can see there in the diagram. And these sacs are actually, it is thought that the main role of these sacs is to actually decrease the speed of transit of the digestion. So it gives more time for the cecum and colon to actually ferment that feed and to absorb nutrients from that feed. So the main function of a large intestine is water absorption, will be number one. And also, of course, and not less important, is the site where microbial fermentation will take place. In most species, in a more or less degree, there is the existence of microbial fermentation. So there will be some fiber fermentation occurring in that region with the production of volatile fatty acids, and also gas, and also some vitamins like B, the B complex vitamin will be actually synthesized mainly in this region. But of course, this part of the gastrointestinal tract has a limited absorption because we're talking about the terminal length of the gastrointestinal tract. So we won't have much time for absorption. We will see how some animals actually compensate that with coprophagia. And of course, the main exception for this limited absorption, which actually are absorbing in important quantities, again, is volatile fatty acid, propionic, acetic, and butyric acid, and water. So let's have a look at the different type of digestive system we will find in animals. We have monogastrics, polygastric or ruminants, and monogastrics, which present a large cecum. And in this case, of a typical example of monogastric will be swine, the pigs, horses, and chickens. In polygastric, we will have cattle, goats, and sheep. And as monogastric, we will have mainly the horse. So in this table, we can see how the length of the small intestine actually varies and changes according to the animal considered. And then what we, there is a kind of interesting ratio here that show that it is made by the small intestine length considered by the body length of the animal, and also the transit times of digesta in the small intestine. So we can see in the case of a cattle, they have a ratio of 20:1. That means small intestine to body length. And they have by far the more slow transit time of 75 hours, which of course, if you think about it, is logical considering the important fermentation process that takes place in the rumen. Then we have sheep and goats, 27:1, horse, 12:1, so we kind of much less, with a 38 hours transit time. So we can say that the horse is somewhere intermediate between a ruminant and a monogastric. And then we have the pig, 14:1, the dog, 6:1, with 23 hours of transit time. So we can say 24 hours to make it round. And finally, the cat, with only 13 hours of transit, with the much smaller small intestine length to body length ratio. So as you can see that as we move towards the carnivores, normally the length of the small intestine actually goes down. It's less, it's kind of shorter. So let's talk about monogastrics. Well, this is a typical diagram of a monogastric. Most of the time we use as a model the pig. We have the esophagus, which communicated with the stomach through the cardia, and then the stomach with the small intestine with the pylorus, followed by a small intestine of about 60 feet or 2.5 gallons of capacity of volume. And then we have the ileocecal valve, which is actually a valve, kind of an sphincter that actually controls the passage of food from the ileo to the cecum of the animal. Its cecum in the pig is of about 9.5 inches length, half a gallon capacity, and finally the large intestine. These are also other diagrams. We can see the digestive tract of the dog and the cat, with following pretty much the same distribution than the pig as a monogastric. And in case of birds, things change quite a lot because it's a completely different digestive tract, characterized by the presence of a crop where mainly food storage takes place, followed by a proventriculus, sometimes referred as a true stomach or glandular stomach, followed by the geyser, which is a kind of food grinding pouch. And we will see that there is even the presence of grit that actually contributes to the mechanical grinding of feed. And then the other thing that is very characteristic of bird is the presence of two cecums, two cecas, so a pair of cecas that end in the large intestine, followed by the cloaca. And the cloaca is also unique for birds. So we have here in this diagram a gastrointestinal tract dissected of a bird that in proportion they have quite an important pancreas, and you can see the liver as well. It's kind of important, and the pair of cecums, which is kind of unique. So the poultry are characterized by this mouth, which has a beak, no teeth, and with this keratinized beak, they actually are able to pick up small particles from the ground like grains, and it secretes there is production of saliva, followed by the crop, which can be defined as an enlarged area of the esophagus, which actually holds and moistens the ingestion, but the crop is not the esophagus, it's considered a different anatomical structure. In some avian species, fermentation takes place in the crop, and sometimes that's the way the chicks actually feed from the crop of their parents. And we have Then the proventriculus, or the glandular stomach, which is characterized by the production of gastric juice, mainly pepsinogen and chlorhydric acid, like monogastric, like a pig, for example, we saw, and followed by the ventriculus or gizzard, which is characterized by having a thick muscular wall. It is actually fulfills an important mechanical function, reducing the particle size of the feed, and contains the grit, kind of little small stones that are acquired by the animal that actually contribute through the muscular contraction of this pouch for the grinding of the feed, followed by a small intestine. And then we have the ceca and the large intestine. As I said before, in poultry is characterized by the presence of two cecas, a pair. It is, as in most species, it is the main site of water reabsorption and also some fiber digestion, following by a cloaca, sometimes called the vent, which is a terminal portion where most of the excreta are going to be eliminated from the gastrointestinal tract. And we will find a mix of feces and urine. And if you remember, birds are able to excrete uric acid, which is almost non-toxic compound, and it's also a very important and significant way of saving water. We have here diagrams of different hindgut fermenters. We have, for example, the horse, which has the mouth completely different teeth if you compare it with the cattle, and you have the prehensive teeth, lips, and tongue. As I mentioned before, the lips are important for the selection of the different grasses the animal will be feeding. They produce saliva with almost devoid of enzyme, no enzymes, up to 10 gallons per day, and followed by a stomach, which is relatively small. It's not very big, smaller than other species. And the horse is characterized by having no gallbladder. It doesn't mean that they do not produce bile. They produce bile, but the bile is being secreted and excreted from the liver without the existence of a gallbladder continuously. And you have to bear in mind that the physiology of the horse tell us that these are animals that are continuously feeding. If you see them on a grazing condition, you can see that most of the time the horse is grazing. And that's why there is actually no need for the bile to be collected on a pouch to be released only 2 or 3 times of the day, like will be the case of other species. This is a diagram of gastrointestinal tract of the horse. We have a large intestine, which is really important and represent about 60% of the total capacity of the gastrointestinal tract, where we will find a very active microbial population that ferments different feed, producing, like we saw before in cattle, volatile fatty acid, acetic, propionic, and butyric, and also it is an important place for the synthesis of microbial protein, which is going to be digested and utilized by the animal in its use. But it's going to be a limited use because remember, these animals are post-gastric fermenters. So the main site of absorption, which is the duodenum, is not present. And for this reason, we say that they normally have a limited use for the fermentation products produced by the microbial population. Here I have another diagram. You can see in proportion, the huge cecum and the colon, mainly the colon. And this is also one of the reasons why these animals are so prone for colic impaction and colic problems. In this diagram, we see what we call the anatomical reference or topography and topographical anatomy, showing the actual place or the region where the large colon occupies in the horse. And remember, the cecum is normally located on the right side of the abdominal cavity. So this is, I know it's not exactly the same structure, but comparing with the cattle, when cattle are bloated, we will see swelling on the left flank. In the case of a horse, when they are bloated for some reasons, we will experiment that. We'll see that bloating mainly occurring, that swelling on the right flank of the animal. And here we have the different capacity of the cecum and colon on the hindgut of the horse. You can see that the cecum has a capacity between 28 to 36 quarters, while the large colon has 86 quarters. So it's really an important volume. And of course, hindgut fermenters have a much greater capacity for fermentation, both the cecum and the colon. And finally, I want to talk a little bit about sickle fermenter, which is the case of the rabbit. We have this particular habit of practicing coprophagy, and they produce two types of feces. One of them are hard that are produced during the day, the droppings we normally see in the cage of a rabbit, and then some soft ones that normally they actually perform the coprophagy only during the nights, and these feces are called cecotroph. They contain mainly content from the cecum, as a different from the hard droppings that actually contain the content from the whole gastrointestinal tract. Okay, these are only from the cecum, and normally they are re-ingested as they are produced by the animal. You see the picture there, a rabbit eating its own cecotroph. And by this mechanism, the rabbit, what they do is they reabsorb those normally nutrients and vitamins that will be lost because of the, we can say, regional situation of the cecum and the colon. So in the cecum, so these animals will benefit more with the microbial fermentation, and they will acquire the vitamins that are mainly produced in the cecum that otherwise will be irremediably lost. Here we have different diagrams showing the cecum size according to the different species. In the case of a swine, we see that is a very small cecum. In the case of a dog, it's almost non-existent. Then in the rabbit, much bigger because it's a sickle fermenter, as we saw. And finally, the equine, a huge colon and also quite a big cecum because they are, of course, colonic fermenters. And with this slide, I finish this first part of the lecture of anatomy and physiology of the gastrointestinal tract and comparison of the gastrointestinal tract between species. Next half, we will talk about rumen. Thank you very much.